Hydrophobic interaction chromatography, theory and principle. Hydrophobic interaction chromatography is a powerful technique for the separation and purification of biomolecules. Hydrophobic interaction chromatography is widely used for the separation and purification of proteins in their native state, as well as for isolating protein complexes and studying protein folding and unfolding. Hydrophobic interaction chromatography was initially termed as salting out. Hydrophobic interaction chromatography utilizes the reversible interaction of protein and the hydrophobic ligand for the separation of protein mixtures. Proteins separates out based on the increasing order of hydrophobicity. Proteins containing hydrophobic and hydrophilic regions are applied to a hydrophobic interaction chromatography column in a high salt buffer. The salt in the buffer reduces the solvation of sample solutes. As solvation decreases, hydrophobic regions that become exposed are adsorbed by the media. The more hydrophobic the molecule, the less salt is needed to promote binding. Usually a decreasing salt gradient is used to elute samples from the column, in the order of increasing hydrophobicity. Sample elution may also be assisted by the addition of mild organic modifiers or detergents to the elution buffer. Hydrophobic interaction chromatography theory. There are three major theories explaining the mechanism of hydrophobic interaction chromatography. They are salting out and hydrophobic interaction theory, thermodynamic theory, and surface tension, van der Waals forces theory. Salting out and hydrophobic interaction theory. The elution or precipitation strength of an ion is described by the Hofmeister series. Small, highly charged ions are strong precipitators, whereas organic acids and bases have a more stabilizing effect in the presence of proteins in solution. The term chaltropic refers to the ability of the ion to produce chaos in the water structure. Anti-chaltropic salts, such as ammonium sulfate and sodium sulfate, expose hydrophobic patches on proteins by removing the highly structured water layer which usually covers these patches in solution. As a result, hydrophobic residues on a protein molecule can interact with the hydrophobic ligands of the matrix. Salts can also reduce the solubility of proteins by shielding charged groups, which normally keep proteins apart in solution. When the electrostatic charge on protein molecules are shielded, the molecules can easily interact, form aggregates and eventually precipitate. The solubility of different proteins is reduced to different extents by salt addition. Chaltropic agents disrupt the intermolecular forces between water molecules, allowing proteins and other macromolecules to dissolve more easily. Chaltropic agents interfere with stabilizing intramolecular interactions mediated by non-covalent forces, such as hydrogen bonds, van der Waals forces, and hydrophobic effects. Thermodynamics theory. Hydrophobic interaction chromatography. Thermodynamic theory of hydrophobic interaction chromatography directly relates to Gibbs free energy equation. The phenomenon of increased interaction in the presence of salting out ions is explained by a higher gain in entropy when water is transferred from the surface of a nonpolar molecule to the bulk of water. In hydrophobic interaction, the entropy change is of greatest importance. If hydrophobic molecules such as aliphatic carbon chains are immersed in water, the monolayer of water molecules in contact with a carbon chain will be in higher order than those in the bulk of the water. If two or more hydrophobic structures come together, the surface that has to be covered by ordered water molecules decreases, some water molecules join the less ordered bulk water, and the entire system gains in entropy and thus decreases its free energy. This state will be favorable for energetic reasons and thus promoted. Surface tension, van der Waals forces theory. A third theory suggests that van der Waals forces are responsible for the hydrophobic interaction in hydrophobic interaction chromatography. This is supported by the fact that These forces should be increased as the order of water increases, in the presence of salt. Hydrophobic interaction chromatography media. In hydrophobic interaction chromatography, media is described based on ligand, ligand density, available capacity. Available capacity is the actual amount of protein that can bind to the media. If flow rate is included in the defined conditions, then it is called as dynamic binding capacity. Dynamic binding capacity is based on these factors: salt concentration, flow rate, temperature, and pH to a lesser extent. The characteristics of good hydrophobic interaction chromatography matrix are high binding capacity, physical stability, chemical stability, and the matrix should be inert. Hydrophobic interaction chromatography ligands. Ligands used in hydrophobic interaction chromatography media has alkyl or aryl groups. Alkyl shows pure hydrophobic character, while aryl shows mixed behavior. Ligand attachment to the matrix in hydrophobic interaction chromatography is done through glycidyl ether. Binding and elution in hydrophobic interaction chromatography. Binding is done at high salt concentration, 1 to 2 molar ammonium sulfate or 3 molar sodium chloride. Elution is performed by reducing the salt concentration. Elution of proteins from the hydrophobic interaction chromatography will be in the increasing order of hydrophobicity.